Hello, everyone. Welcome to the Introduction to Image Analysis session using Imaris today. This is Kevin Frischmann from the Application Support team in the U.S., along with my colleague Arvonn Tully, also from Application Support in the U.S. And today we'll be walking you through an introduction of how to use Imaris in general for 3D image visualization and analysis. It's a bit different from earlier sessions in that we're trying to leave a significant portion of the webinar for a Q&A session. So if you have any questions along the way, definitely jot them down if you'd like to address me directly, or feel free to type them right into the chat pod. All of your questions will be going into the chat pod anyway, so put them in at any time. Arvonn will be answering your questions as we go, and for any questions that would be better answered with a visual demonstration, we'll hold those until I finish the initial presentation, and we can show you an answer to your question rather than just tell you. With that in mind, we'll try to keep things moving fairly quick for the introduction to Imaris, sort of tutorial portion in the beginning. It will be recorded, so if anything is moving too quickly, you can always come back to the recording and, of course, ask questions later. So thanks again for attending, and with that, we'll go ahead and get started. As I said, for those of you who may be very new to 3D imaging, we'll have just an overview of the concept of how we assemble a 3D image data set, a little bit about Imaris as a software package, what it's comprised of for anyone who's brand new to the product, and then I'll do a brief live presentation of just some pretty basic tasks to get people started on the overall workflow, and then we'll go ahead with the Q&A session where you can ask some specific questions to follow up on what I've shown, or just anything at all if you think it might be possible to do it within Imaris or otherwise. So Imaris is definitely optimized for multidimensional data of the confocal imaging type, regardless of the method for acquiring a confocal type stack. That doesn't matter too much to the software, but also other imaging modalities, really anything that acquires a set of pixels in 3D, which are normally called voxels. So each 2D section has some pixels, and when we extend that to the third dimension and acquire a stack of 2D images, we can refer to them as voxels, since the sampling volume occupies some specific volume. It's a volume element. And those individual planes, each one through one method or another, is able to restrict the acquired image data to only a particular plane of focus or some spatial plane by another method if it's not based on focus. And then we know the exact position where the imaging system was focused when that image was acquired, and so we have some depth data about each pixel in space, and we can assemble those into a stack of pixels that gives us our volume image with now voxels in it. And so we have the usual X and Y spacings. If we have a properly calibrated system, we know how far apart each pixel is from its neighbor, and if we have a properly calibrated stepper or whatever the focusing mechanism is, we also have some information about exactly how far apart each focal plane is. And so now we have image data that is registered to a particular point in 3D space, and we can then reconstruct what that three-dimensional object would look like based on that information about the image that was acquired at each point in space. And of course, if you repeat that cycle of acquiring a stack of images over and over again and watch how things change, then you have so-called 4D or 3D over time. As far as Imaris goes, what we do is take the image that's already acquired. So it's not something that hooks into your acquisition system, but we have many, many file readers so that you can easily get your then acquired data into Imaris. And the core module, if you want to call it a module when you're just starting out, is just the visualization. So we can reconstruct something visually that looks like the object that was imaged. And we can adjust the visualization and use different styles of slice rendering and volume rendering and shadow projections and surface rendering and something called spots to detect point-like or blob-like objects and render them as spheres or ellipsoids. But it's all just visualization. There's no information extracted about the intensities or the volumes or the shapes or the distances and so on. If you add to that Measurement Pro, um, what you see here are some objects detected as surfaces, and there could be any number of ways to do that. We'll talk a little bit more about it later. But in the core module, we could see those surfaces and render them and rotate them and take snapshots and make movies, but we would not be able to extract information about the volume and intensity and so on, shape, etc., of each one. So Measurement Pro adds the possibility for each object that you segment or detect or extract from your image, you can get a bunch of statistical information out of that particular volume and shape and so on. In addition, there are things like distance measurements, as is shown in this screenshot here, and some other tools that are tied to the Measurement Pro module for manual contouring. Now, if you are doing time series imaging, you might want to add the track module, Imaris track, to your configuration, and what that does is allow you to extract additional measurements, and to get to those measurements, you also need some tools to connect the different time frames together. So you have a collection of objects at time frame 1, and you have a collection of objects at time frame 2, and the job of a tracking algorithm is to determine which ones are the same object, which ones are new objects that just appeared, in which cases did objects disappear and potentially split or fuse and so on. All of those things are related to a tracking algorithm. So first there is the process of doing the tracking, and then of course there are all kinds of time-relevant measurements. Once you follow an object over time, you can of course look how its intensity or volume changes over time, but you can also look at time-specific things like velocity and direction of travel, acceleration, total displacement over time, and so on. The colloq module is specific to looking for similarity of structure between two different color channels to give you some idea of how significant the overlap is between two signals, and then extract the positions of those overlaps, get some information about their correlation of the intensities and so on, and even build a new channel that you can then go back and detect surfaces and spots or filaments, etc. from. Filament tracer is primarily used for neuroscience applications, but as the name implies, there are other potential applications in the form of anything that is somewhat filamentous or line-like or tube-like. As with all of the other modules, it traces in 3D space and can handle the time series as well. And here again, the method of detection is a bit different. Sort of like when you add the track module, there are some additional segmentation tools added to actually extract the features of interest. With filament tracer, there are algorithms specific to filament-like objects to help you better represent the object that was imaged in an accurate way, so you get an accurate segmentation and therefore accurate measurements. And of course, there are filament-like measurements associated with that, having to do with the length of each segment and the number of branching points and the sizes of the branching points and their distance from a beginning point, volume and so on, density of spines attached, etc. So those are all of your filament-specific measurements and many more that would be added. In Imaris Cell, you have the only case of one of the segmentation tools that is a multi-component segmentation. So in this screenshot here, you see that there are some cells detected, and those cells have nuclei, and within those cells are some other subcellular components. The software refers to them as vesicles, but they could be anything point-like or blob-like within the cell. that you would label their position with these spheres. And then when you do this multi-compartment type of analysis, you can start extracting information, one, about relationships between the different compartments in the cell, different subcellular components, having to do with how far are the vesicles from the plasma membrane or from the nuclear membrane or the center of the cell, et cetera. And also, it provides a more convenient way if you have a field of view containing multiple cells. The segmentation algorithms are going to separate the results on a per cell basis. And so it becomes more convenient to get information like the volume of each cell in the field, and then look at that from different angles, the nucleus to cytoplasm volume of all of the cells in the field, or the number of vesicles for each cell in the field. And then, of course, if you have a time series, you can look at how things change over time with respect to those measurements. And once you start extracting measurements from any of those surfaces, objects, or cells, or filaments, or spots, the Vantage module allows you to go in and look at those measurements in a plotting interface or in a gallery view, and arrange different axes based on the measurements that can be extracted within Imaris. And as you can see in this particular image, one option is to render the components of that object directly into the scatter plot. So here you're looking at different segments of the filament that was detected, and instead of rendering them in their original shape and spatial relationships, each segment has been rendered as a separate point in the plot with its actual shape, and we're looking at the straightness versus the length versus the mean diameter, and showing what the actual object looked like that is represented by that point in the plot. That's not the only way to do it. You can do traditional point plots. You can even render volume rendered objects with a varying intensity that look a little bit more like the original fluorescence image. And if you do that, then maybe you can get some idea of how the intensity varies without plotting the intensity or something like that. And then there's the Imaris XT module, which it's a little bit hard in a quick description to fully convey what it is. It is a programmable interface, would be the simplest description, but it is also a package of add-on plugins, essentially. We call them extensions that come with Imaris that were developed by BitPlane to extend the functionality. But essentially what it does is allow Imaris to communicate with some external program so that you can write custom functions for Imaris. And that could be in MATLAB or in Java or Python, or anything that can bridge to MATLAB or Java or Python as well. And so you can take what we've written and modify it, or write something from scratch, but it allows you to take the software in a direction that's much more specific than maybe a commercial entity like BitPlane can take it. So those are all of the modules that are part of Imaris. And then there's also this somewhat attached to Imaris component called Imaris Batch. And basically that allows you to take some analysis parameters, a set of commands. It doesn't necessarily have to be analysis. It could be some visualization settings or some image processing, and just apply that to a larger number of files instead of having to open them up in Imaris one at a time and apply those settings to each one and save it. So you can batch that up and just let it run overnight if it's a large set, or over a shorter period if it's handled. There are a couple of external software packages that are not really part of Imaris but do exchange data with Imaris. One is Auto Aligner for aligning serial sections so that you can reconstruct a 3D volume from a series of images that were not acquired in an aligned manner. And then also the AutoQuant deconvolution software package does send data back and forth with Imaris to make the workflow a little bit more convenient. In that case, we don't produce the AutoQuant software, but we do support it, and it is somewhat integrated with the Imaris interface. And for those of you who aren't familiar, deconvolution software basically takes you from the top image down to the bottom where we're removing, not necessarily removing, actually there are multiple options. Ideally what you want is to reassign some of the out of focus light back to its origin, the object that generated that out of focus light. And so essentially you get a bit better resolution even with images that are already confocal. Particularly along the z-axis as is shown at the bottom here. Okay, and with that, we'll switch over to the live demo, and I'll give you a little bit of an overview of the workflow through Imaris. Whenever you are loading your images, there are a large number of formats supported, and we would strongly encourage you to use the original format that came off of your microscope or other imaging system, because that's the best way to maintain the proper calibration regarding the space between the voxels, and if you're doing a time series, the time between each frame. If you have some questions about whether a format is supported, you can just ask, but if you have Imaris in the open dialog, if you take a look at this list, this should give you a pretty good idea of what you should be able to load. And if you load one of these formats and it doesn't load the metadata properly, please let us know. It's always a changing field, and we're not always the first ones to know about a change in a file format, but in theory, all of these should work properly so that if you load one in, and look at the image properties under the edit menu, the voxel size that's represented here under coordinates should be accurate, assuming that the system was properly calibrated. And so this is the information that Imaris needs in order to calculate things like distances and volumes and speeds and so on. And it's very important to make sure that this information is accurate when you first start, because it's not possible after you've gone through an entire process of analysis to go back and change this, because there are too many variables about the way the result could change as you start adjusting space and time post-analysis. So always check this whenever you're trying out a new file format to load into Imaris, or if you update the software on your acquisition system, which could lead to a change in the file format. You want to just verify that everything's being read in properly. And if you're working with a time series, there's this all equidistant button. When you press that, you get a pop-up showing the interval between time frames. If you don't know the size per voxel, but you know the field width, you could enter in a min value of 0 for X and 17 microns for Y, and that is the width of the entire field along the X-axis, and you could do the same for Z. It's fairly normal to have a small negative value here. It's representing the corner of the voxel rather than the center as being the minimum extent, so don't worry about that if you have negatives. Once you have the image properly calibrated, so if it didn't load in correctly, you can of course type in the right values here as you start. And if you have to do that, it's probably a good time to then save the data set, and you probably want to save it into the Imaris format so that anything that you are doing in Imaris can then be preserved with each time you save the file. The other advantage to saving in the Imaris format if you have large data sets is that they are potentially much quicker to load into our software than some other formats. So if you are working with some extremely large images, it makes a lot of sense to convert them to IMS files, and there's even a batch converter that you can use to do that if you have a series of files that take a while to open and save. Once you have the image loaded in, if we switch over to the basic slice view here, the first one is showing us each of the individual focal planes that were shown in the PowerPoint slide earlier. And then over here, the display adjustment window, which I'll extend a bit, shows that we have 4 channels, and we have some channel visibility options. We can completely hide a channel, or we can adjust the contrast using these 1, 2, 3 arrows, which are linked to the boxes down here if you want to just type in a value as well. If you press the auto button, it's going to set to the min and max range of the histogram. So that is often a good starting point. And then also it's often a good idea to look at one channel when you're adjusting the contrast. And so if we bring down the max value, all of the brightest intensities are going to get brighter still, but at some point we start to lose contrast. We don't want to go too far with that. Also, if there is some background in the image that we don't want to view, we can raise the min value and set, in this case, the black cutoff a bit higher so we don't see the background. And then the middle one is your gamma adjustment if you want to have a somewhat nonlinear intensity on the display. Important to note the name of this box is display adjustment because that is all you are adjusting, the intensity on your display, including any snapshots or movies that you capture. So it will have this level of contrast if you capture a snapshot or a movie, but if you are detecting some objects, it's not going to have any effect on the size or shape or number of objects that are detected, and it is not going to have any effect on the intensity measurements that are extracted, strictly for display and rendering purposes, so don't worry about it affecting the results. You can as well click on the name of a channel here, which is a shortcut to the image properties and the page for that channel. If you would like names displayed in the display adjustment, you can type them in here. There's some additional information for some applications could be useful. And then if we want to change the color, we can do so right there. And there are also mapped colors available if you would want to have some kind of multicolor lookup table. You can apply that as well. You can create your own lookup tables or just use the built-in ones. Now there are some other basic views. If you click on this arrow here, you can get to them next to the slice view. The section view gives you a 3-up kind of view here, where we have the XY, XZ, and YZ, and we can reposition where it's slicing through in each dimension. You can also do extended sections and drag these arrows, lines apart to show a thicker section in the opposing view. And then if you want to look at individual slices, they're here in the gallery. And Easy 3D is honestly rarely used, so I'm not going to get into the details of that right now. When you switch to the Surpass view, the default behavior is to build a scene containing these 4 objects, a Surpass scene folder that contains a light source, a frame, and a volume object which is set to maximum intensity projection mode. Each of these checkboxes are also visibility checkboxes, so we can hide the volume rendering or hide the frame. At the moment, the light source isn't doing anything, so we can only see it if I select it in the list, and now it's available for interaction with the mouse. And if you have a group that you uncheck, it hides the entire group. You can change the order of these objects by dragging them, and if you have a scene with multiple groups, you can change what group they're a member of, and every object you can double-click on to rename. In some cases, the order is actually important to how the visualization turns out, but I don't think we'll necessarily have time to cover that today. There are additional objects either in the toolbar here that you can add to the scene, or if you go to the Surpass menu, those same objects are represented here. Some of them are strictly for visualization, others are for feature detection and analysis. The 2 we'll be talking about today are the spots and surfaces objects. Besides the volume rendering, you have some basic slicers you can show in this view as well. And they can be oriented in different directions, and you can have more than one of them, and a number of other visualization tools there. So if you wanted to have multiple slicers, if you wanted to have oblique slicers at some odd angle, all of that can be set up. And then if you want to get rid of any objects, you can either hide them or select them just like you would files in your operating system, and then click this delete trash can icon here to get rid of them. For each object that you select in this list, there are some properties down below. So for the light source, it has color and fixed to data set. Frame has quite a bit more. So it's going to change the color of our frame object. or if we want a grid or not, and so on. And then the volume object has different rendering modes. And for each one, the display adjustment is probably going to be tuned a little bit differently. You can see the blend mode might not be your favorite if you don't know how to set it up, because essentially everything looks black initially for a lot of fluorescence images. So I just want to quickly review how to set that up. The maximum intensity projection is convenient because it requires almost no adjustment to get a good high contrast image where you can see a fair amount of structure in every channel most of the time. But it doesn't give you any information about relative depth or position of neighboring objects. So we can't tell if these red points are at the front or the back of the scene, really. If we go to blend mode, we have this added property of opacity that you see in this slider here, right now just adjusting the blue channel. And that allows us to see the relative order of things in the Z depth and get an idea of relative positions. But it's a bit difficult to adjust because all of the channels look black. So when you're using blend mode, turn off all of the channels but one, and initially just adjust them one at a time. You have your opacity here. Oops, wrong channel. So you have to select the channel by clicking on it, and you'll see the histogram down below changes to match as well as the name. We can adjust the opacity there, and the max value, and the min, and the gamma. And then just repeat that. Off with that one, on with the next. Try to clear as much of the background without getting too aggressive initially. And usually you'll want some opacity adjustment, but it depends on the image. And eventually you end up with something that looks quite a bit better than what we started with. But the blend mode does take a little bit more work. So if you just want a quick and easy rendering, it's not always the best choice, especially if you have a lot of channels to adjust. But it does give you some more information, so I personally prefer it if I'm using it as a visual reference to guide some object segmentation, get an idea of how accurate the segmentation is and so on. Once you've got them individually adjusted, then switch them on. You've gotten rid of most of the gray and black, and you can tweak the contrast for each channel individually. But now you can see which of the kinetic cores are up towards the top of the stack and which are toward the bottom and so on, without having to do a full rotation of the image. If you'd like to capture a snapshot of some of your images, there's a snapshot button here. And here we specify the path to save our snapshots for the current data set. They'll be saved in TIFF format. You set up the parameters. You see a preview here. And then finally you click Do Snapshot, and it will write to that path. If you're allowed to write there. And then it increments the name so that you can set up multiple snapshots and keep clicking without changing the name. And you'll just get a numeric series of snapshot names. And it's a toggle button, so if you click snapshot again, it goes away. And now if we wanted to detect the number of kinetic cores in one of these channels, we could go ahead and do that. So I'm just going to hide the other channels for the moment and make the red channel transparent, a little bit less opaque rather. And we can do some spots detection. And the reason I would choose that is because they are fairly compact, point-like, blob-like, and of similar sizes. And that is an ideal scenario for the spots object. And once I know that, once I'm familiar a little bit with Imaris, the first thing you do then is add spots to the scene. And for some objects, instead of settings initially for visualization, you get a creation wizard down here, an object detection wizard. And if you had used some other settings previously, you could save them and recall them, or you can start from scratch. It is possible to set up a 3-dimensional or 4-dimensional region of interest if you only want to segment a smaller area. And you specify the channel, the approximate size, most people like to just look at the slice view and zoom in a bit with the middle mouse wheel, and then just click a couple times on the image when Imaris is asking them to estimate the size of some objects. And it's looking around 0.3 or 0.4. You can see that size displayed over here in the distance box. And there is an ellipsoid option. If you have some Z stretching of small objects, you might want to turn that on and potentially adjust the ratio. That will help get better centering and counting and measurements, etc. And usually you'll want background subtraction enabled. And then there's a quality filter, which is more or less a signal to background, where you can exclude some objects as noise or include more. And you can also manually add and remove some spots. There's an automatic threshold, which it varies whether it will work on one image or another. And there are some other approaches the support team can help you with to do automatic thresholding if the initial value does not come up the way you'd prefer results-wise. There are other filters as well if we wanted to exclude objects close to the image border or apply some other filter to include or exclude objects. We could do that and have a whole series of filters here to get a subset of the initial result. And then finally, we have some spots objects. You can see their ellipsoid nature there, and they have some statistics associated with them if you have the Measurement Pro module. Some basic things like number of spots, but then also a larger number of measurements. And there will be even more than what you see in the initial list here. If you click on the Statistics Settings button down in the bottom left, it will take you to all of the possibilities for the spots object and what you can switch on or off there. The other interesting thing is that you can click into a row in the table and see which object that is, or you can click onto the objects here if your pointer is in select mode. There are two pointer modes. Navigate, you use the mouse just to rotate with the left mouse, zoom with the mouse wheel, or pan while holding down the right mouse. When you're in select mode, that's where you can interact with the objects in the scene. And if you hold down the control key, you could select several objects and maybe see statistics about only the current selection or export that to a file and so on. Once you have some statistics, you can export them to an Excel file or a text file using these buttons down here, and there are different collections of statistics, just the currently displayed ones, or all of the statistics that are currently enabled in the preferences, and so on. Now, if you have some more irregular objects like the blue channel that you wanted to measure the volume of, you could detect those as surfaces because bots would not be so well suited for that. And then from there, you can get information about the volume of that, and you can maybe split the spots into two groups if you have the right modules for those that are inside or outside. And for each object that you detect, whether it's spots or surfaces or cells, when the intensity data is extracted, it's for all channels present. So for each red spot, you will see how much blue signal and how much green and how much white is within that spot. And same for the other way around. There's, of course, positional data, and you can do more with that as far as how far are all of the red spots from the white spots, for instance, here with the kinetochores and Van-Actin in this image. For the surfaces, I'm going to just bring up a different image that's a little bit better to demonstrate some of the other features. Okay, so here we have some objects. They are of fairly similar size. They are fairly blob-like, but their shape, their elongation is not along the z-axis. And that ellipsoid spot tool is really only able to deal with the z elongation that occurs when you're detecting objects that are close to the z-step size or the z resolution of your optics, where they tend to get stretched out a bit for optical and/or sampling reasons. Here, there's an arbitrary orientation to the elongation, so ellipsoid spots would not really help us. and it could be difficult to get the right information out of these chloroplasts in this case, detecting them as spots, because the sphere would fill part of the object rather than the whole thing. If we needed the intensity information, for instance, we would have to go to the surfaces object. And again, we get a wizard here. If I wanted to exclude the edges, I could enable a region of interest. And then often it's easier when you have a fairly thick stack to switch the camera type over here temporarily to orthogonal. And that just makes it easier when we switch to select mode to grab these handles without having to rotate the data set. At a larger image, we could do multiple regions of interest here. Again, we need to specify which channel contains the objects. Here, it's asking us for size again, but it's for smoothing, and that's why it asks for surface area detail level. Objects smaller than this are going to be lost, so you want to be careful with that, and you would want to measure them. If you're not sure, often the default is a good starting point. If you have a variety of brightnesses, you can enable local contrast. And specify roughly how large the objects are. That will basically help even out the contrast at this step where we have to apply a threshold. And again, we can take the automatic threshold, or we can adjust it manually. Here, though, if we wanted to count or locate these objects or measure them, you can see that they're a bit closer together than the spatial resolution allows for them to be detected as separate objects. So then what we need to do is use this split touching objects and estimate the object size. Like the spots, you do need to have objects of fairly similar size for this automatic splitting to work. If automatic splitting can't work, then you can always manually split objects. And then if we have overlapping spheres, those should be merged into one if they have a significant overlap. And it gives us an idea of how the splitting will work at this stage. This is essentially our spots detection combined with the surfaces. And then you can see some split lines resulting here. And then we have a final result. And there are some measurements associated with that, objects that we've detected. If we want to take a look at these in a sort of different arrangement, we could switch over to the Vantage view. Initially, you'll just see everything color-coded for the default measurement of surface area, and everything is in its original position, really. I'm going to switch the camera back to perspective here so we can see a little bit more depth. And then there's, again, this idea of creating something using a wizard, where I click on Next and fill in some parameters about the type of Vantage I want. Gallery can be interesting because it orients everything with the long axis along the same direction, and then you can sort them by some sort of value. In this case, it's by volume, and it can give you some idea quickly of which objects should have been 2 when they were detected as 1 based on their overall volume. And you can still interact here and select objects. And then if you go back to Surpass, you can see exactly where those objects are in the original positions as well. And that goes for the other Vantage views here, so that if we want to create a scatter plot based on area versus volume, we could do that. And we could add dimensions to the plot as well, changing the color and assigning that to intensity, for instance. And then, of course, you can also adjust what the color map is, or the size of the axes, and so on. And here again, if you want to click on some objects in the plot, you can still access those. And then once you finish the wizard, there are additional options to fine-tune the plot visualization here in the Vantage view. And if you have time series, you can do measurements over time and have a time axis and so on and so forth. Okay, so that's a quick overview. As I said, there's many more modules that we will hopefully cover in other sessions, but happy to take questions now and see how things go. I'm going to switch over here and see what we've got. And Arvonn, if you want to just pipe in any questions that you thought would be good to demonstrate as you were answering them, feel free. So it'll probably take me longer to find them.
Kevin, I'm putting a couple off to the right-hand side.
Gotcha.
That I think would be good to talk about.
All right.
Actually, if you want to take a look at, Lonnie had a question about showing snapshots of an image at an angle. Do you want to show them the MATLAB camera manager? I don't know.
Yeah,
or shall I do that?
So if you want a specific angle for a snapshot, it sounds like that is the question. There's a couple different approaches you could take. The Imaris XT programmable interface does have a interface to the camera in Surpass. So if you go to the image processing menu, you'll see a number of MATLAB-based functions if you have this module, and there are a couple called camera functions. One is set the angle of the camera, and if you don't have MATLAB, you'll see the Windows command prompt pop up while the MATLAB compiler runtime launches, and then a MATLAB window will appear. And here you can directly control angle, elevation, and azimuth of the Surpass camera prior to taking a snapshot. You can also, if you want to set something up interactively, you can take a look at that window and say get angles from Imaris, and it will tell you what the current angles are. Now you can also call up this camera manager extension, and what this allows you to do is create some camera configuration file. Now there will only be one of these files where you can save multiple views if you like. So you only want to browse someplace first where you have write access and save your Imaris camera file there, and then you can save something into that file and repeatedly do that. And then if you want to recall a view, click on load. Well, I should change the angle first just to demonstrate. You can click on load and grab that oblique angle name that you stored, and it will set up that particular view. Now keep in mind that the camera view that you get depends somewhat on some absolute coordinates, and so if you're going across different data sets, you would want to make sure that the range of min and max are pretty similar across the different data sets that you're trying to apply the same camera settings to. The other thing you can do, just if you want to enter in a specific rotation and not necessarily store them for future use and apply to other data sets, you can sort of abuse the animation view. So down here are our global interaction buttons that are always visible. If you click 100%, it sets the rotation to 0 and then fits it to the screen. And if we enable the animation toolbar here, normally what you would use this for is to create a movie. And the most basic one would just be to make a rotation by clicking this 360-degree horizontal, and then each of these vertical lines is a frame in your movie, and currently there are 100 of them specified. We can look at each frame in the movie or drag through them. And with a 360-degree rotation, What it does is tell the camera to travel to a point that is rotated 90 degrees around, and then go another 90 degrees. And so these are increments of exactly 90 degrees around your data set if you would want to capture those. If you want something other than 90 degrees, under rotations, click Custom, and this is referring to the axes of your computer monitor if it's set to screen, or the axes of your microscope and your data set if it's set to world, as I call it. If I want to rotate around the y-axis of my screen 45 degrees, I can click OK, and it inserts a keyframe after whichever one was previously selected. I have to go off of that keyframe and back to it again, but now it's rotated exactly 45 degrees from the previous keyframe. Keep that in mind that it's a relative rotation. So if you want it relative to 0, first click 100% and set the zoom where you want it. Then do your custom rotation of however many degrees, and it's going to rotate relative to the earlier position. So if I want to go another 10 degrees and get to 55, I could do that here by typing in 10. And I get another keyframe that's rotated a little bit further. Okay. What else do we have, Arvonn?
Can we, so we had some requests for doing tracking and how to measure speed and displacement. That was a good application to show.
Okay. Certainly a common application for Imaris. So we'll load up, obviously, a data set that has a time dimension, for starters. And this one happens to also have a z dimension. Now these objects, they don't elongate too much, and so if you are interested in tracking their displacement and their speed and so on, the spots object is going to be most likely the better choice because it's a little more robust to errors compared to surfaces. But if you needed to know the shape or the exact volume of each of these objects, you might have to go with surfaces, which could complicate the segmentation process a little. If you have a time series, this checkbox won't be grayed out anymore when you add a spots object, and it will be checked by default. Here again, you have to estimate the object diameter. The default is just a multiple of your voxel size. So whatever 10 voxels is, Imaris is going to guess that your objects tend to be around 10 voxels wide. So it could be way off, and don't worry too much if it is way off your actual object size. In this case, they're probably closer to around 6 microns if we look at them in an individual slice. So I'll go ahead and put that in. Here, they're large enough that there's not a huge amount of elongation due to optics, but this data set has been downsampled a bit, and so you do have some elongation just from undersampling. It's going to be a problem in general with the segmentation, so your results in Imaris are generally going to depend on how good your image acquisition is, somewhat not surprisingly. And at this stage, we have the quality filtering, and it looks pretty good. Of course, if you have a time series, you'll want to drag along the time slider that just appears along the bottom whenever you have a time series to check and see how the segmentation is doing at other time frames. If you are wanting to get the most exact segmentation possible, it does allow you at an intermediate step to add and remove spots. So if you put the pointer in select mode, the cursor gets a box around it, and you can roll the mouse wheel to adjust your estimated object size. Now, it's not going to do ellipsoids, unfortunately, so it's not the best fit for manual segmentation, but it will try to center this in 3D space where you click based on the box size that you used for the object estimate. So it didn't place it at the top or the bottom of the stack. It's pretty well centered on the center of intensity for that object. You can also click this recenter button, and it will shift even closer to wherever the local maximum is in 3 dimensions. You can also allow the detection to fail for a small number of time points as an object moves along and still get an accurate result because there's something called gap closing. There are different tracking algorithms to choose from. We can't really get deep into the details of them today, but most of the time the default autoregressive motion algorithm for most applications works the best, but you can talk to the support team about the details of that. The maximum distance is referring to how far can an object possibly travel between 2 adjacent time frames. If you don't know what that is, often it's useful to look at the frame object and see what the spacing is there and or change the spacing there. So if we step that down to 10 microns and move through, we can pretty easily spot that there's a fast mover in this region of the image, and it's probably jumping between 10 and 15 microns based on that grid spacing. Autoregressive motion does tolerate some variation or some exceeding the maximum distance, and the larger we make it, we will be somewhat more error prone. So I don't want to go too far with it. Maybe we'll do 12 microns. Gap size is number of time points that an object can disappear and still be picked up. So if we look down at the bottom right here, there's a cell that was detected, and if I go to the next frame, it's gone. That's a gap of 1. The next frame, it's still gone. That's a gap of 2. Next frame, that's a gap of 3. In the following frame, it comes back. So that means that Imaris will go back to these 3 frames and run the spots detection again in a very small region of interest that is close to the predicted position for that object at each frame, and try to find the spot even though it was a little bit below the initial quality threshold. Here again we have a gap of 1, 2, and then it's actually gone, so we don't need to worry about closing it. Now if you don't check this box, it won't run that spots detection on the gaps. It will just leave them as gaps and try to make a connection between, for instance, time point 10 and time point 13. In between, at 11 and 12, there will be no measurement data. You will have some information about the overall displacement of that track, but you won't have information about the instantaneous speed or the intensity or the volume, etc. at frames 11 and 12, or whatever they are if you uncheck this. It varies with the image, basically whether you expect the object to be detectable at the gaps, whether you would turn that on or not. Then finally, you actually get some tracks. By default, it's going to filter out very, very short tracks that are only a couple of frames long. And you can, again, add other filters if you wanted to look for tracks of a specific duration or straightness and so on. At this point here, you have your tracks and you have some information regarding the tracking. So here the acceleration vectors are currently shown. You have the speed, that's instantaneous speed at each frame, and of course the XYZ position. You have the total displacement, which is referring to this line here. If we go to the settings tab, we can change the track visualizations. So what's the length of total displacement, and then you also have the displacement squared there if you're looking for directed versus random motion, track straightness, track speed variation, and so on. So you have some numbers having to do with how it's traveling from one frame to the next, and some numbers about how it traveled from the first frame to the last of that particular track. There's a number of other visualization options in there. If you want to show this dragon tail, if you have a high density of tracks, that can be useful so that we're not filling the screen necessarily with track lines. You can make it a bit longer, and basically the object then builds its track, and at some point starts erasing the tail end so that it doesn't get too long. And if you have any errors, there's an opportunity to do some editing. Here it looks like there were 2 cells detected instead of 1 at this frame, and so we got a break in this track. If I select this point here, you need to be on a certain tab to be able to select objects. I'm going to go to the Edit tab, and we can, rather the Track Edit tab, and we can choose whether the mouse selects an entire track when I click, or just an individual spot, a single time frame. I only want 1 cell at this frame, so I'm going to select an individual spot, and then delete it.
Oh, Kevin, can you be sure to show drift correction?
Yes, I can show drift correction. It will be a little bit odd on this data set. It will be kind of a forced application since there doesn't seem to be any drift here, but it will get the message across nonetheless. So we can definitely show that example. Now I'll go back to the track edit and select entire tracks. And just to make it clear, the dragon tail is on, so there is a track continuation here as we go. So I can now go to my track editing, select this track, hold down Control, and select the other track. And if I want to, I can do this Show Selection Only checkbox, and it's showing me in this view here only those 2 tracks. I could draw a connection holding down the Shift key or just press the Connect button, and then I get a continuous track for that object, and all of the statistics are adjusted accordingly, and so on. Now, if this object was not supposed to be moving, but its motion actually represented the movement of the stage or the organism that contained the object that we were tracking, we could do some drift correction. And the way that works is you select that track, you tell the software that this object should not be moving. If you want to do an average of multiple tracks, if there's some noise, which there typically is, you can hold down Control and select multiple tracks for objects that should not be moving, and then click the correct drift button. In this case, it doesn't make sense to average these 3 tracks together, and it won't be so clear what it's done. So I'm going to select 1 and say this object should be stationary, press correct drift, specify if there is only XYZ translations from one frame to the next, or if potentially the scene, the stage, or whatever the object is can rotate. You need at least 2 tracks if it's a 2D image for rotational correction. And if it's a 3D image, you need at least 3 tracks so that we can figure out the plane of rotation. And then these other options here, crop largest common region, new size equal to current, and include entire result. If you look at the preview down here, it's telling you that the new image will occupy the pink space if the drift had gone from the bottom black rectangle to the top black rectangle here. So crop largest common region, if our field of view was initially over here and ended up over here, we would end up with a data set that was much, much smaller. Now, if your drift is much, much smaller, that's not such a big deal. So it depends. In this case, we would get a very small data set because the travel of the drift is very long. Include entire result is actually going to fill in. It's going to pad the data set so that we don't lose a single voxel after applying the drift correction, but the data set gets larger. And if the path of drift is significant, the data set can get significantly larger. So keep that in mind. The middle option is just a happy medium between the two. So the final size of the data set will be the same as when you started, but it might remove some voxels at the edges, and it might add some black voxels in the corners, depending on how the drift moves. This final checkbox here is just going to adjust all of the tracks for the objects that are supposed to be moving. And it varies. I'm not going to get into the details of why you would pick that or not, but it's fine if you leave it on most of the time. And actually, I'll do include entire result so that it's a little more clear what has happened. Obviously, the tracks look very strange now, but if I hide them and crank up the contrast here, you can see that the entire volume now is moving in the opposite direction of where that cell was moving. So it has subtracted the motion of that object from all of the other objects in the image and actually modified the content of the data set to change its position at each frame, to subtract the distance that that cell traveled at each frame. And then basically what we're seeing now, if you look at that cell right there, it doesn't move at all. And we're seeing how everything moves relative to that supposedly stationary object. So that is drift correction in an odd sort of way. What else do we have? You want to show one, Arvonn?
Well, I've got a couple questions about distance transform. So do you think we should do a distance transform demo?
Sure. Popular feature.
Also have some filament tracers. So maybe try to think if I have any good filament tracer measurements. So let's see, do you want to make me presenter? Let's see, or no, I have to make myself.
Yeah, I'll just stop sharing my screen. I guess I could have given you remote control, but if you'd rather share your screen. Okay.
Yeah, I'll do it on my screen. Okay, so someone else is going to have to take over typing duty. I think I caught up to everybody.
Okay, I'll keep an eye on the chat.
Let's see here. I'm going to pick a little bit easier image. Just give me one moment. So I'm going to go over the filament tracer tool. And normally we do a little bit different image, not the usual pyramidal cell, because I'm sure everyone has seen that many, many times and is tired of seeing that. So to use the filament tracer object, what we're going to do is go in here and select the filament object, and then we can add that. Now, I know there was a question about if you have multiple cells or multiple filaments in your region of interest, in your image, what you can do is select region of interest. And what you can do with this is you can go to the select mouse, change the mouse to the select mode, and you can actually change the region which is going to be used for the processing. I recommend changing the camera type to orthogonal first. Makes it a little bit easier to grab these sections. So you can use this to restrict where the software is going to look. So if you do have multiple cells in your field of view, you could select just one. This is a relatively small image, so I don't need to do that. So I'm not going to select a region of interest. If you're using like a conventional neuron that has a cell body and processes, use the autopath method. If you're looking at something like blood vessels or non-hierarchical images or non-hierarchical structures, you would use this threshold method. I'm going to show the autopath method. I'm going to go to the next step. Then what you do is you set the largest diameter, the starting point. We can do a quick measurement. Someone else had asked about doing quick measurements. If we go to the slice mode up here, so I'm going to go up here to the slice mode, and then I can scroll up and down through the image. I'm going to find this cell body right here. I'm going to zoom in. I'm going to select left click and then left click again. And I'm actually going to change the color really quick to make it easier to see. I'm going to go green. So I just select the channel name, which is my neuron, and then we can change the color. There was also a question about what is how to reuse colors. You can either use a mapped color, so we could go over here, select a particular mapped color, or in the base color you can type in a value. So if I know I want to use a green, I can type in 0 for red, 0.5 for green, and 0 for blue, and this will always make the same color green. I think Ben Abrams had that question before. So now that I have selected, I can just left click and then left click again. And this will show us the approximate size here over on the right hand side, 13 microns. So I'm going to use that as my rough starting size. I might go a little bit bigger because the long axis here is a little bit bigger. So I might do like a kind of halfway in between, maybe 17 microns. Then it's going to ask the thinnest diameter for the actual processes of the dendrites, not the spines, but the processes. So let's go something like that, half a micron, maybe a little bit larger, about a micron maybe, 1.5. I'll go with that. See how that goes. And so then what Imaris is now going to do is it's going to go through and identify and highlight these structures, and go through, let's see here, go to navigate. And what we can see is Imaris has placed a starting point in the correct location, and then it's placed these seed points along these paths. And so now we can adjust this to add more or less points if we feel that's appropriate. We also have the option to remove seed points around the dendrite, and we can adjust this value. And so what this does is it clears out seed points around the starting point. Basically, that improves the detection. So sometimes if you don't have this, what you will see is that you'll get a lot of false, kind of incorrect connections there. So we like to use this to kind of clean that up right around the dendrite starting point. Sometimes you can make it even a little bit larger. You see there's some thick branches here. We might want to set this a little bit bigger. Oops. And that'll help clean up those connections there. The other option that we have, we don't often show, is remove disconnected segments. And so you can see we've got some seed points out here. Now Imaris will try and connect all of these regardless of how far the gap is. So if we use a remove disconnected segments option, what this will do is this will basically make a mask around our structure and give us the option to exclude objects which are too far away. So like this one right here, and we can exclude that. We can exclude these. So let me show you how that works. So now we've created a mask over this structure. I'm going to set this a little bit lower to make sure that everything is included. And then what we can do is we can set a maximum gap length, so basically a distance to exclude these structures. So I think 30 is a bit large. Probably go with about 15, half that. And then we'll go to the next step. So anytime you have a gap of more than 15 microns, Imaris will exclude those from the tracing. So we'll let this finish here. Are there any questions that we're missing? Looks like Kevin's got it.
Arvonn, there was a question about the distance between spots and density of spots in a defined area. That might be a good one for a demo.
Oh, yeah. Let's see here. I guess I can... Yeah, of course, the filament tracer can take some time to calculate. Let me see if I can start another instance of Imaris.
And maybe I can ask you, Nathan, who asked that question, as well as Serena, or maybe they have different answers. Just go ahead and type it into the chat. Would you be interested in distance between spots that are in the same color channel, or distance between spots that are in different color channels? Because the answer is then somewhat different.
Yeah, we can do this quite quickly using the PK2 sample. Here, let me see if our filament, okay, good, our filament is ready for the next step. So as I change, this is a threshold for determining the diameter of the filament, and as I bring this up, you can see where the paths for the filament have been created. And you can see these filaments out here have been excluded, but some of these separated structures were a little bit were closer, and so those were included. So that really should not have been included. So maybe I need to use a smaller value. So like some of these smaller ones were included. So that was it's probably about 15 microns right there. So that's why it was not excluded. If I'd used a smaller value like 10 microns, that probably would have been excluded. So now, but I can also go in and show a little bit of manual editing and how to use the filaments. So then setting this threshold allows us to determine the diameter of the filament along each of the points here. And that's really important for doing the spine calculations. So how do we do that? We can either let the software choose an automatic threshold, which is in theory the best way to do it. The other thing that we can do is we have a choice between shortest distance from distance map or the approximate circle of cross-sectional area. So what the shortest distance from distance map does is it looks at this mask and finds the shortest distance along the cross-section of this mask to this center line here. So that's one way to calculate it. Typically, your XY cross-section will be the shortest distance. The other option is to use the approximate circle of cross-sectional area. So what this does is along each point, which is roughly 1 per voxel, along each point of this structure, you will see the software will basically do a perpendicular projection to that and find out what the cross-sectional area of this distance mask is. And then it will use that area and calculate a circle who has the equal area and use that circle as the diameter of the filament at that point. So we can go and see what that result will be. Generally, we recommend using the shortest distance from distance map for the cross-sectional area for dendrites, and then the approximate circle of approximate area for the spines. But depending on your data, one may work better than the other. You can see it works pretty well, but sometimes we run into a few problem areas. But again, we know these are that I didn't set the limit far enough there, so that's okay. We have the option to do spine detection. And so what we do here is we select the source channel, set the size of the spine head, which is about 0.5 microns in this case, and then we set the maximum length. Obviously, we don't want spines that are 25 microns long. That's probably incorrect. So I'll put in a little bit more reasonable value of 5 and let the software find those spines. And interestingly, it didn't find any. Let me double check something really quick. Oh yeah, the voxel spacing is too large. I'm going to have to put a little bit bigger size and say like 1 micron. That's probably. This image doesn't really have enough resolution to determine spines, so this section will be kind of incorrect, but it's more just to show you how it's supposed to work. So there we're kind of fabricating some spines. Again, we can adjust the threshold to have more or less spines. I'll go with automatic. And then Imaris is going to calculate the shortest path of maximum intensity from each of these spine heads back to the dendrite. This does take a little bit of time. So while we wait for that, I'm going to go over to this other image and let's find how close these channels, these two sets of white and red spots are to each other. So I'm going to go ahead and add some spots. Let's find those white ones.
Just interrupt you briefly, Arvonn.
Sure.
So each person had a different question. One wants distance in the same channel, and the other wants different channels. If you could quickly show both options.
Oh yeah, sure. Well, I'm going to start with the. I guess we'll start with the. Well, I'll set up two sets of spots, and then we'll show them how we can do both. So these spots are about, these white ones are about 0.2, 0.3 microns. So we'll do 0.3. So we'll find these white spots first. Looks like we've done a fairly good job of finding them. So I'll go in and name these. So I presume that everyone's familiar with how to use the spots tool, at least the people who are asking this question specifically. Then I'll also go in and add some red spots. And so again, we're going to say these red spots are about the same size, 0.3 microns, 0.3. So I'll add the spots object. I'll tell it which channel, then I'll set the diameter. Go to the next step, and I'll add a few more. There we go. And we can adjust the quality threshold here, or any of the other measurements that we're interested in. So now what we have is we have some white spots and some red spots. So, and we'll name these. So the first tool that you might be interested in is kind of knowing, there's a couple ways that we can get some clustering information here with the red spots. First we can, and this all assumes that you have MATLAB installed and the Imaris XT interface. So the first thing you might be interested in is using the spots to spots closest distance function. So again, I'm going to select the red spots. I'm going to go to the Tools tab, and then I'm going to go to the spots to spots closest distance. So I'm going to click that. I hope I have MATLAB set up and running. And good, I do. And so now what this is going to do is it's going to select the results mode. So I'm going to actually select spot statistics and spot radius. And this is just going to run on this particular spots object. Let's say OK. Select the distance mode. I'm going to do center to center. And what this is going to do is it's going to create a new spots object, which shows the basically how close these spots are to each other. So now if we go into the spots distance objects, we're going to have some extra measurements here. Distance max. So this is the maximum distance from this spot to any other spot. We also have the distance mean. So this is the average distance. So if I select, say, this spot here, it will show me the mean distance of all of the other spots to this spot is 5.4 microns, 5.39. I can also look at the distance minimum. So what is the closest spot here? So 1.26 microns is the next closest spot. So now if I look at this spot, I can see that its closest micron, or its next closest spot from the red spots is 3.28 microns. So this is one tool for determining kind of what your clustering is like between these different spots. So you can see these bigger spots are further away from the other spots. So these two, so this is showing you by default the how close the other spots are, what the next closest spot is, what's the minimum distance. So that's how that's displayed. And also we add extra statistics in here. So that's one tool if you just have a single class of spots. The next tool that you might be interested in is using the split spots function. And that's right here, split spots. And what this does is, again, this is only on a single set of spots, one spots object. And so what I can do is I can run this split spots function. And again, this is through MATLAB. And split spots into subgroups which have a distance greater than a particular threshold. So it's basically going to try and cluster these spots. So I want to cluster these into 2-micron clusters. I'm just picking kind of an arbitrary value. I'm going to say OK. And then so what Imaris will try and do is try and find the clusters of spots that are roughly 2 microns apart from each other. So now this has created a new spots group object, which has the different clusters. So you can see one of these clusters, they're all evenly distributed and quite close to each other, 33 spots in that cluster. But then the rest are in other clusters. So that's another tool that we have. The other tool that people typically use is the co-localized spots. And so what we can do here, this requires 2 sets of spots. And what we do is we go in, doesn't matter which one we select, and we're going to go into the red spots and select co-localized spots. And so what this MATLAB function is going to do, you're going to select 2 spots to co-localize, in this case white spots, red spots. I'm going to say OK. And then it's going to enter a distance threshold. So in this case, I want to look for spots which are less than 0.5 microns. And this would be the center-to-center distance. So if we look and we know that these are about 0.3 microns, maybe it'd be better to do, if they're within one radius, so that'd be what, 0.15 microns, so we'll do 0.2 microns. If these spots are within 0.2 microns of each other, then that will count as co-localized. So now when we select that, we're going to get two groups of spots, okay, down here. These are co-localized spots. The white spots co-located and the red spots co-located. And we're going to see which ones are co-localized and which are not. And if we go into the top of the group here, and we select that and we look in these statistics, it's going to show us in the overall statistics overall for this group object, the number of spots. And if I make this a little bit wider here, I can just left click and drag. It will show us that we have 3 white spots co-located and 3 red spots co-located. So you can even go in and drag your red spots and your white spots in there to kind of get an overall picture that we've got 3 white spots co-located out of 5 white spots. And we've got 3 red spots co-located out of 51 red spots. So that kind of covers the basic tools that we provide for doing some spots to spots closest distance. Did I miss any, Kevin? Or I think I really covered it there.
Yeah, I think you're pretty well covered for the spots distance.
The distance transform is the other one. Yeah, I forgot about that. Should I do that? I'll go ahead and do that now, I guess.
Yeah, go ahead and get that done. It's pretty similar.
Now, so there, actually, I did leave out one additional method. Oops, wrong one. So another tool that we can do is if we want to find out, let's say, how far these red spots are from the white spots, we can use the white spots as a reference. Now, in order to use a distance transform, what we're going to need to do is we need to go edit, change data type. And we need to tell, and I'll do this a little bit more slowly, edit, change data type. And then we can go in and set the type from, in this case it's an 8-bit image. We're going to set it to 32-bit float. Now the reason that we need to set this to a 32-bit float is that distance measurements are fractional, and 8 and 16-bit images only allow integer values, 1, 2, 3, whereas a floating point image allows us to have fractional values, 1.25 microns. And so that's going to be very important whenever we do a distance transform. So I just went ahead and said OK. I'm going to select the white spots. I'm going to go to the MATLAB Tools function, and then I'm going to select distance transformation. And from the spots, it's going to do a distance transformation from the center of each spot. Now, just so you believe me, what we just did, I'm going to go to the slice view, and I'm going to turn on the channels, and I'm going to turn off the original channels, and I'm just going to leave this distance to white spots. I'm going to click that and change the color. I'm going to go to a mapped color just to make it very clear what we're doing here. Change it to jet. And then I'm going to adjust this color range so you can see where the spots are. So we've calculated, let me go to section view. For each of these spots, okay, you can see we now have a distance map, and it shows how far every voxel is away from each of these spots. So what this allows us to do, okay, is we can now figure out how far each of these red spots is from the closest white spot. So now I can go to red spots. Now, because we added this channel after the red spots were created, what I'm going to do is I'm going to go to the statistics, and I have to actually force these statistics to be rebuilt. So I'm going to go to the spots. I'm going to just toggle one of these statistics, and this will force all of the spot statistics to be rebuilt. Now that those are rebuilt, I can actually go in to the detailed statistics. So this is the red spots detailed statistics. And I can select a specific value, which is intensity center channel 5, okay, because this distance is channel 5. So now this new distance channel is the distance from one of these red spots to the closest white spot. Okay, so we could actually sort this and go up here and find this red spot over here is in fact its closest, it's 8.4 microns to the closest white spot, which looks pretty reasonable considering how far away it is. And if I go to the next one, which is this one, this is 5.6 microns away from the closest red spot. So you could imagine sorting this and looking and seeing, look, aha, one of these has a value of 0. So this is effectively in the same location, and of course this is co-localized, as we would expect. So we could look here and see that in fact several of these are co-localized, very, very nearly co-localized. So what did we say? We had 3 red spots co-located, right? So now let's go and take a look over here, and it looks like we had 3 that are less than 0.2 microns, which should correlate exactly to our 3 red ones that are co-located. So these 3 were co-located there, and if I turn this on, we can see that these 3 are also, oops. Yeah, so those are the same 3, which is no surprise. So it's just another way to get those values. So this could be very interesting if you use, say, like average values. We could go in here and see what's the average value, how close on average are the red spots to the all the white spots. So now if we look in the red spots statistics detailed average values, we can go to intensity center channel 5, and we can see that on average they are 3.7 microns away from the white spots. So that's just a really nice way to do some measurements. And you can see what their min, 0, max, 8.4, standard deviation, the median, etc. So that's just kind of some nice tools that we have for dealing with 3-dimensional object-to-object measurements here. So let's go back to the, go back here. We're going to do the spine, same question here for the spine measurements. I'll go ahead and just finish that. The exact values aren't terribly important here, just giving you an idea of what the process is. So now we've gone ahead and done our filament recreation. And I know some people had some questions about shoal analysis and how we might want to go in. And so what we can do to get the shoal measurements is to go here to the statistics, go to detailed, and then we're just going to go ahead and select shoal analysis. And if I don't see that it's turned on, it should be under this filament section. One thing you can do is you can actually restrict some of the measurements, and we only want the filament measurements. So it should be here, and if it isn't, there we go, filament number of shoal intersections. So this will tell you, I'll make this a little bit bigger, this will tell you the value, so at 0, where are all the intersections. So of course everything is highlighted, and then if we select 2, this should show you all the ones that are at 2 microns. Not terribly interesting, but if we go and select something out like here, around 34, yeah, at the radius, I'm sorry, the radius of 48 should show you all of them. One thing I can do is to highlight just those, I can do a mouse selects point, and this should actually select just those points at a particular distance. And it's selecting everything. Okay, well, made a mistake. Now, there was a question about how to change that shoal radius. The default is 1. If we go to Edit, Preferences, Advanced, and we go to the Surface App Filament here, Preferences, Advanced Surface App Filament. We look in here, we can actually find the shoal sphere's resolution, and we can change this value to something that might be more conventional, like 10 microns. Say OK. That will force the rebuild. Now the shoal radius will be calculated. Here's the radius, 0, 10, 20, 30, and this will show you the shoal value for that radius. So I think that that kind of answers that. Let me see here. Got that. Got that. I'm going to return to sharing. Stop sharing. Let's see here. What are the next questions that we can answer?
There was a question from Marie that I'm not sure if you follow it better than I do.
I'll take a look at it here.
And then there was a question as well about filaments if you don't have a soma, and sounds like they were pretty well satisfied, but if you want to show a couple semi-automatic modes for drawing, it might be good as a quick demo.
Yeah, sure, I'd be happy to do it. Let me pick a different image here, and then I will share. I can do the. It's just kind of a dendrite, but I want to do a different image. So just give me 1 second to prepare. Okay, I've got a good image here. So here's an example of a dendrite which does not have any cell body. So how would we approach this? So again, we're going to add a filament object, and I'm going to show you kind of, I would say that kind of the preferable method, but then I'm going to go through and do it another way. So I'm going to use the creation wizard. And now what I'm going to do is I'm going to set a starting point. It doesn't actually, now this image is not particularly calibrated, so please don't be distracted by the voxel spacing here. I think it's just set to 1 voxel. It's not yeah, it's saying that's 6.4 microns. It's not quite correct. But for the purposes of demonstration, this is fine. So I'm actually going to set it to a very large size, like say 20 microns. So something that a size that does not occur here. And then I'm going to set this thinnest diameter to something very realistic. So in this case, it's about 5 pixels or 5 microns, saying. Set this 5 micron value. And then I'm going to go to the next step. Now, these starting points are totally wrong. In fact, they're so wrong, I'm not even going to use them. I'm going to set these values to something that's totally bogus because it doesn't matter. And what I'm going to do is we can go in here and I can say Shift right click. So the mouse has to be in select mode, and I can do Shift right click and it will place a seed point right there. Then I can set, now there's a bit of a problem, and I'm going to adjust the contrast here a little bit to make it easier to see. I'm going to change the color so we have a little bit more contrast. And then I'm going to change to blend mode just to make it really, really easy to see where these seed points are. And you can see there are a ton of seed points. We don't really want that many because these are spines, and we don't want to track those. So I changed that by opening up the display adjustment, clicking on the channel name, setting a color. Then I can close that. And then I also went to the volume object and switched to blend mode. This is just my personal preference. Obviously, if you're more comfortable using the MIP, you can do that, but I find blend works really well here. Then I'm going to go back to the filaments object. And I don't want all of these seed points, okay? In fact, I really only want them along the main dendrite. But the problem we have here is that this dendrite is roughly the same size as all of these seed points. So what I'm going to do is I'm going to set my seed point threshold very high, and then just go in, and I'm going to do Shift left click. I'm going to manually place a seed point all the way at the end here. And I'm going to do a couple along the key points. Now, one thing you'll notice is that the size of my mouse cursor determines the placement of that seed point. So, and again, I'm using the escape key to toggle between select and navigate. And if you get absolutely nothing else out of today's demo, that's the one thing you need, is escape toggles between select and navigate. Anyway, what you want to do is you want the mouse cursor to be roughly the size of your structure. If you have the wrong size, what will happen, and of course it's not going to happen right here, but if you have the mouse cursor at a really wrong size, it's going to place the spot incorrectly. Okay, so you see there, I placed it. It's a really big spot, and it'll just place it in totally the wrong spot. But if you have the mouse cursor the right size, then the spot will be placed in the correct position. And we don't actually need that many spots, many seed points to place this. We can also go through and do Shift left click and delete that one on a spine. So I've got enough spots here. So you see I've just got a few, one all the way down to the tip, and then these will be traced all the way back to the starting point. I'm going to go to the next step. And one thing that's actually nice is that the number of spots is proportional to the amount of time to calculate. So you can see that Imaris has just gone through and quickly calculated the seed point, calculated this filament all the way out to the tip there. And now we can go through and use kind of our regular tools to identify the dendrite. Get the diameter calculation. So this is kind of one semi-automatic method. We can detect the seed points here, so I can go through and say, oh, these are up to 5 pixels, 4, let me get a good size estimate here. Some of these are as small as, I'll go with 4, maybe 3, 3.5. And of course this 50 is quite long, but again, it's 50 pixels. So that doesn't do too bad of a job there. Go to the next step. So this is one way to do it. Okay, so now, and again, I don't really want to go over this too much, but just showing you that it was possible to set the start and the end points manually for the dendrites. It's also possible to do that with the spines if you know that any are correct or incorrect. Okay, so that's one way to do the calculation. Now, there's some other ways. So this was, I would call this AutoPath manual creation wizard. That's one way to do it. Let me show you another way to use a mix of manual and automatic creation to identify this dendrite. So I'm going to add a new filaments object. And now what we're going to do is we're going to skip automatic creation and edit manually. And so what we can do with this is we've got now 4 semi-automatic methods, and well, 3 semi-automatic methods and 1 completely manual method. AutoDepth is very similar to what we did with this AutoPath manual creation wizard. And what we do with AutoPath is we set kind of a filament diameter using the mouse wheel. So I'm using the mouse wheel to adjust the size here. And I do Shift right-click, and this will place a starting point in the image. And then Imaris will go through the entire image and make a map. And once this is done here, let's give it a moment. This is calculating basically the entire data set. And what we'll be able to do, give it a moment, is from that point, we can basically just click into the image, and you can see, right, it works all the way up to right about here. And now if I do Shift left click, it now has traced that structure automatically based on the size that we sent. And now in theory, we can even go back and tell it to do spines. And if I can Shift left click and add spines here. So that's one way to do it. Now, that's if you're interested in doing this manually. That does a pretty good job. Although after we do that, we would still have to do because this is not doing a diameter calculation, we would have to go back, use the edit tool, and do some diameter calculations. The other thing that we can do at this point is that we can take this filament, and I'm going to make it a little bit easier to see here. We can take this filament, and we can actually go to the creation wizard and rebuild the spines. So I'll keep the existing data and click on this rebuild. And this will jump back into the creation wizard and allow you to basically kind of use these settings and use the existing dendrite, but now find spines around this using our creation wizard. So it's one thing we often recommend is that you can, if you have a dendrite, just go in and draw a dendrite by itself and then use the creation wizard to find the spines, because that's a very quick way to do this. Might have to redo the dendrite diameter there, but that just gives you an idea of another way we can do that. So I think we're about done. Are there any other things that we need to go over? Let me go back to the chat pod and see what's going on. It looks like
yeah, I think everything was pretty well covered there. Just scanning through to see if we missed any, because there was a little bit of jumping around. Marie's question was relative to performance, so we can take that up with her later potentially. It's a little bit mysterious to me. It was slowing down with a large background subtraction value for surfaces. Doesn't sound normal.
Yeah, that's normal.
Okay, anyone else with last-minute questions? See some typing going on. If you have any follow-up questions, just go ahead and send them to US support. I'll go ahead and share my screen again. We have some additional information here. So for follow-up, particularly for Imaris XT, you have the Imaris Open, but it is a general discussion forum where customers can submit things and get answers. If you are interested in having something written, a custom function in Imaris XT, you can ask if anyone's available to do it in that community site. And then depending on your region, if you're in the Americas or Europe or Asia, there's different support emails you can send a message to. And then within Imaris, you have the help menu with a number of items there. If you click anywhere in the software and choose Show Help, it will try to load a relevant page in the manual. And that just loads into your web browser. Tree, and I see that page, and of course the manual is searchable as well. In the help menu, you have the video tutorials. That's a link to the website. There's also some local tutorial content, the quick start tutorials. And if you go to the video tutorials, that's actually part of the Learning Center. And it's pretty well indexed so that if you just type in a particular term, you can find some videos and how-tos relative to a particular keyword. You might want to go into advanced search sometimes and hide the items for the multimedia gallery. So they're just snapshots. Hopefully that answers that question. I'll definitely see what we can do about getting the content of the chat Q&A, especially for things that weren't recorded into the video. But aside from that, I think we're all done here. Thanks so much, everyone, for staying and for joining. And personally, I think it was a great turnout and some good content, good questions. So hopefully we'll be doing this format again. If you're not subscribed to the newsletter, you can go ahead and do that on our website, and you'll be notified of any upcoming webinars of various types, whether they're training or applications presented by user and so on.